So this presentation is work done by a postdoc, Steffan Hansen, which all of you probably know. And just like Karain, he got a job and ran away, so I'm standing in for him. And this is sort of a report on initial studies that we're starting on the biological control of PSHB. And so currently there's multiple people involved here. So PSHB has a lot of hosts, as I mentioned. It's also associated with 3 symbiotic fungi. We only focus usually on Fusarium because that's the pathogen in the trees. But the other 2 fungi are also there. That's the food for the beetle, Paracremonium. We don't really know what that does. And then also, we've noted that these beetles are often associated with mites, small little mites that climb on the beetles. As the beetles fly between trees, the mites travel with them. So we had some ideas of what to do with them. So we know that microbial biological control is often quite effective in IPM programs and might also be so for Uvalacea fornicatus, for PSHB. So we decided on looking at 2 biocontrol fungi, Trichoderma, as an antagonist against the fungi, the food for the beetle, and then Beauveria as something that can actually kill PSHB. And it's shown in lab experiments that Beauveria can kill PSHB quite easily in a petri dish, not in a tree. And then we've also noted lots of phoretic mites, and we've thought that we could possibly use them in this biological control system as taxis for the biocontrol fungi in the system to get the fungus inside the galleries of the beetles. So we're not there yet, but we're playing around with the ideas. A couple of phases we need to go through. The first phase, we're sort of at the end of that now, and we need to make sure that this Trichoderma fungus is actually a good competitor against the food for the fungus. So we've tested it against a whole bunch of the PSHB symbiotic fungi. We tested it when there's an unoccupied resource, so there's an open resource, and whoever comes first wins. Tested when the resource is already occupied by something else, so that's probably what happens in the galleries. The food for the beetle is already there. And then also played around with the inoculum levels. All of this currently is only done in the lab, so the next step would be to try and do this in a more sort of natural system. So the Trichoderma turns out to be a really strong competitor against all of these fungi. It overgrows them, it kills them quite easily. It also kills the biocontrol fungi for the beetle. So we'll have to have different approaches when we actually apply these things. So that's good news. On a petri dish, it works quite well. Still, we don't know what's going to happen in the tree yet. Looking at the phoretic mites on the beetle, so Steffan ran around, chopped down some trees, and looked at the mites on the beetles. Don't worry about the names. There's a whole list of mites that were collected from PSHB, and we are focusing on 2 of these that we often find on the beetles, and that we also can grow in the lab, colonies of these things, so they can multiply it. So it turns out these 2 the mites feed on the fungus that the beetle also feeds on, so we can breed it quite easily in the lab. The first one is Tricharopoda monroei, which is a native mite, known from Africa from other similar beetles in similar systems. So this is a mite that climbed onto PSHB when it got into South Africa. And then there's an undescribed Histiogaster species, which we have no idea where it comes from, whether it's native or not. But both of these we have in colonies now in the lab. So phase 2, which a master student is starting with now, is to actually start looking at if we can use these fungi in a more assimilated system, natural system. So building on work from Nell et al., we know that the Beauveria can kill the beetles in the petri dish. They tried it to do it in a tree, and it had 0 effect in the tree. So we want to see, go through the steps to see where the problem lies. So we're going to test the entomopathogenicity of some of our own Beauveria cultivars or isolates that we got from dead PSHB beetles in the field. And then we also want to see that Tricharopoda, the antagonistic fungus, whether we can actually get this fungus into the colonies, into the tunnels, on colonies that we have on artificial media for the beetle. And then also what the student is looking at is to see if we can use these mites as vectors of these fungi. So we're testing the ability of these two mites to feed and reproduce on our biological control fungi, and it turns out that both of them can do that. So now we need to see how many spores they can carry, and will they carry the spores into the tunnel systems of the beetles. So it's good news so far, but we'll have to see what happens once they reach the colony. Do they successfully transfer the spores, and can they cause issues there? So there's a whole bunch of people involved, a new postdoc, new master's student. Steffan was also luckily still involved in this. And yeah, lots of people to thank, and still a lot to do, so watch the space.